Thank you, and I'd like to thank the organisers for the opportunity to present our work on the Clarity trial, and I present this on behalf of 11 centres and the research nurses, investigators, and of course patients recruited throughout the UK over the last 18 months. Chronic lymphocytic leukemia, or CLL, is the most common hematological malignancy, leukemia in adults. It occurs in about 6 patients per 100,000 are diagnosed with this disease. And the conventional therapy for CLL is chemoimmunotherapy, although the vast majority of patients will relapse after chemoimmunotherapy, and until recently the treatment options were limited, with most patients dying of the disease. The last few years have shown a rapid development in our understanding of this disease, particularly that this disease is a driven disease with all patients having proliferation of the tumour cells, with the pathways now being worked out through B-cell receptor signalling, and also every patient virtually having an abnormality of apoptosis, so the leukemia cells don't die, leading to the accumulation of the disease and resistance to treatment. Both of these pathways have led to novel agents which have been recently approved as single agents for the treatment of CLL. Ibrutinib was approved is a targeted treatment against bruton tyrosine kinase, which was approved in 2013 as a single agent both in America and in Europe, and is approved for all lines of therapy with CLL. It's clearly had a major impact on the outcome of patients with this disease, showing overall survival advantage in many trials over the last few years. However, ibrutinib does not eradicate disease, and patients remain on treatment indefinitely or until progression. More recently, venetoclax has also been approved both by the FDA and EMA for resistant patients with CLL, so the indications are slightly different and slightly wider in Europe. This drug has a very rapid effect, which actually leads to tumor lysis syndrome, which is very unusual in CLL, but also leads to the eradication of minimal residual disease in some patients with a single agent, which we know from chemotherapy trials is associated with prolonged survival. The Clarity trial is a feasibility trial where we recruited 50 patients with relapsed refractory CLL who'd failed chemotherapy or with porous disease who had failed often targeted therapy into a combination study of ibrutinib and venetoclax, so targeting the two main pathophysiological pathways of this disease. The key endpoint, because MRD is associated with better outcome, is the eradication of minimal residual disease at 12 months, with the possibility of stopping these therapies rather than having indefinite treatment. And I'll show you the key secondary endpoints of 6-month MRD in the blood and marrow. Of course, we're also interested in the toxicity of this regime, and we have a variety of exploratory endpoints in this phase 2 trial. The study, we treat patients initially with ibrutinib to debulk the disease. They get 2 months of ibrutinib monotherapy, which is usually well tolerated. We do bone marrows before and after that 2 months, and then at 2 months we add venetoclax. We have an escalation for the first month to abrogate the tumor lysis syndrome, and then bone marrows performed at 6 months, 12 months, and 24 months. And in this trial of 50 patients, I think we've missed 1 bone marrow in all the patients, so that the patients have been very compliant with the treatment. If the patients are MRD, minimal residual disease, negative in the blood and marrow at 6 months, they stop therapy after 12 months, 6 months of the combination. If they're negative at 12 months, they stop therapy after 24 months, because our modelling suggests that's the most efficient way to get a prolonged, potentially indefinite remission. This is the group of patients. We recruited 54 patients. The majority are male, as we see in all CLL trials. They've all failed treatment, but they've had a median of 1 prior line of treatment. 20% of it got porous 17p, and a quarter have 11q lesion, which again is a porous feature in chemotherapy. The majority of patients, 81% of patients, had failed or previously been treated with the 2 most commonly used chemoimmunotherapies, most effective, FCR or bendamustine rituximab, with some patients being treated with a targeted treatment, idelalisib. I should have said that patients were not permitted into the trial if they'd been exposed to either ibrutinib or venetoclax previously. In terms of the toxicity, we saw, in terms of general adverse events, we saw neutropenia, which we see with venetoclax, and some GI toxicity, which is largely grade 1 or grade 2. The particular AEs of interest, because we know these drugs have some side effects despite being targeted, we saw some bruising, so some subconjunctival hemorrhage and some bruising. Neutropenia, which is associated with venetoclax, which responds to GCSF, as we had in the previous talk, and really otherwise very acceptable toxicity. There was a single case of tumor lysis syndrome, which is similar to the 3% we're seeing with venetoclax alone, and that was managed successfully. It was actually in my site, and the patient re-escalated back onto treatment and is doing well. 7 patients had GCSF that has been reported. In terms of response, the key secondary endpoint is MRD eradication. We only have data out to 6 months for 38 of the patients where we have CTs, clinical assessments, peripheral blood, and marrow. And of the patients, 37% are MRD negative in the peripheral blood, 32% are MRD negative in the marrow, and the trophine biopsy is normal in the vast majority of patients. In addition, if you look at the subsets of those patients who have failed chemotherapy and those who have had prior idelalisib, those patients seem to respond at a similar rate to the whole group. In terms of IWCL response criteria, which is a secondary endpoint, 47% of patients have achieved a CR or CRI, and every patient has had an overall response, which for this group of patients is impressive, I think, and that is similar across the patients who are refractory to chemotherapy or have had previous idelalisib. So in conclusion, we've demonstrated that the combination of ibrutinib and venetoclax can be given safely. We've had 1 single case of tumor lysis syndrome, which is what we would anticipate from venetoclax as a single agent, and that was manageable. The rest of the side effects were predictable and, again, were manageable. I haven't shown you the data, but no patients stopped treatment, and only 7 patients have had any interruption in treatment, and only for a few days. We're seeing, even at this very early stage, over 30% of patients achieving an MRD negative remission, which was our target at the 12-month bone marrow stage with this combination. The principle of the Bloodwise Trials Acceleration Program is that we can accelerate therapies and combinations into phase 3 to change practice, and we've already modified our frontline FLAIR trial about 6 months ago to include this combination in a phase 3 frontline study, which is currently recruiting very actively. Thanks for your attention.